Welcome to this edition of the REPROCELL webinar series. Today's speaker is Dr. David Bunton, who is CEO of REPROCELL Europe. He's going to be speaking about how pharma is using human fresh tissues to better predict drug safety and efficacy. David?
Thanks, Bob, and thank you everyone for taking the time from your busy schedules to attend the webinar today. As Bob said, I'd like to give an overview of how pharma is using human fresh tissues to better predict drug safety and efficacy. I'll start with a brief introduction to the company. REPROCELL is a life science company based in Yokohama in Japan, and it's a leader in stem cell and human tissue research. Today I'm going to focus on REPROCELL's activities in human tissues in the drug discovery process, and there are a few particular areas I'm going to focus on which I thought will help to highlight how pharma is using these types of data. So I'll start with the reason why human tissues are used in drug discovery. What is the unmet need? Secondly, I'll give some examples of the over 700 projects that we've done, how they have been used in drug discovery with case studies from some of our clients that have published the data from our research. And then move on to the regulators and how they view human tissue data. What's their thoughts on using human data, and how does it fit with the regulatory guidance documents? And then finally, I'm going to talk about species differences between humans and animals. So REPROCELL, based in Japan, we have 118 people and have sites in US, UK, India, and Japan. Our technical platforms cover many of the types of human tissue research, from drug discovery to 3D tissues through to stem cells and genomics. We also have an extensive biorepository housing many fixed and frozen samples, but I'm going to focus today on the drug discovery using fresh tissues. This is an area which is in growing demand in pharma R&D. It's mainly being used to gain confidence about likely clinical efficacy and to understand the variation between patients, because the tissues are fresh and are coming from the target patient population, and also to identify and understand potential safety concerns. In comparison to animal studies, it's still used relatively infrequently. So today I'd like to explore whether there's scope and good reason to increase its role in preclinical research. So I'm going to look at the access to human tissue, and also how it fits with the objectives of improving our prediction of the likely clinical effects. I'm sure many of you will have seen these slides before in the graphs, before from Kola and Landis and Paul et al. It's a very famous graph about clinical attrition. This describes the main causes of failure in the clinic. Unfortunately, 90% of drugs still fail. It does vary according to therapeutic area. With some areas, there's a bit higher success rate, whereas in other areas, such as oncology, it's closer to 95% failure. So it's very difficult to navigate the entire preclinical, clinical process. There are many reasons why a drug could fail in development. However, we can identify some of the key causes, and the biggest single cause is a failure to predict efficacy. That's partly attributed to the poor preclinical prediction of human responses. We're still over-reliant on animal models or simple cell-based tests, which simply don't reflect the human biology. Another area is safety and tox, and you can see the two arrows around clinical safety and toxicology. Combined, they still account for around 30% of failures. So these are still a major problem, although progress has been made in some areas. Similarly, in pharmacokinetics and bioavailability, this used to be a much bigger problem, and it has greatly improved through increased use of more relevant test systems such as primary hepatocytes and microsomes, but it's still an issue for around 10% of drugs. So there is a clear argument that we need to increase success rates, and we believe that human data used earlier in preclinical testing can help to at least partly address some of these problems. There are other economic drivers behind the use of human fresh tissue. The regulators are encouraging its use in safety assessment. We'll talk about that later in the presentation. Pharma is increasingly outsourcing its R&D, and this actually works well with human tissue experiments because often the providers of access to human tissue or the services around human tissue are quite specialist, and they require companies that can provide those specialist skills and tissue networks. The demand for non-animal alternatives is growing, not just within pharma, but in society at large, and that is putting pressure on pharma to find alternatives, to find more human relevant systems that reduce the number of animal experiments. And more recently, in the past 10 to 15 years, personalized medicine, stratified medicine, or more commonly now, precision medicine, all of which are geared around finding better selection of drugs to get the right drug to the right patient at the right dose. Given the need to identify patients based on their genotype and their phenotype, that type of work in precision medicine is really based on human data. So human tissue research is essential to that area. One of the founders of the most successful biotech companies ever identified this issue of translation from simple systems into more complex human systems very early stage. And he said that you really need to try and use as close to the total physiological system that you can, or you're going to run into trouble. Our objective at REPROCELL is to provide our clients and to use the most relevant model, whether it's healthy or diseased tissue, and really prove efficacy or safety in that physiological system. So how do we actually access these tissues? And apologies for any one of a squeamish nature. Human tissue is, by its nature, it requires us to obtain fresh samples from surgery, from transplant, or biopsy material. This slide illustrates the fact that these are fresh samples. They're not fixed or frozen. They are still essentially living tissues, and they're collected from multiple sites 24/7 and brought to our lab very quickly for research. We've got 14 clinical sites throughout the UK. We have numerous sites, both surgical and also non-transplantable organs. So these are organs which are, of course, offered first for transplant, which is higher priority than the research. But if not available for transplant, then they can be made available for research. And this is a highly valuable source of fresh material, both healthy and diseased, which again can be transported rapidly to our labs in the UK or in Maryland. What do we actually do with that tissue when it arrives in our labs? Well, we do a whole range of functional endpoints on these living tissues. We've shown heart here, but it can be any range of tissues or organs that we study. In all these instances, we are open 24/7 to receive the tissue. It comes immediately to our our labs in the US or UK. And we may conduct a whole experiment then, or we may dissect a specific tissue of interest. In this illustration, we've dissected some small blood vessels. You can see them in the upper right photograph. We then have used those living tissues to study the functional response to a drug. So this is essentially pharmacology in real living tissues. Here you can see a small blood vessel that we have perfused, and we are looking at the effect of a drug that's constricting the blood vessel. And in vivo, this would translate to an increase in blood pressure. A second drug is then dilating that artery, which would translate to a reduction in blood pressure. So these are clinically relevant endpoints that we're measuring in these intact tissues. And although we also do cell-based work, in this presentation, I'm really focusing on these intact living tissues, which could be a blood vessel, but could also be a section of skin or lung or bladder, many other types of tissue that we investigate on a routine basis. So this type of workflow from obtaining the ethically donated and obtained tissue or organ through our lab and then to some functional endpoint is something that we're doing every day here. To give you a feel for the functional nature of it, we have a short video which hopefully you can see this tiny piece of heart muscle that's still beating and contracting in this chamber where we've got physiological saline keeping the tissue alive, and we're able to then measure the effect of drugs on the contraction of that tissue. It just gets across the fact that this is intact live tissue. Now, I mentioned earlier that we will often also look at the other molecular characteristics of the tissue. So we do quite often perform sequencing or transcriptomics on the tissue from that donor. We have bank samples, of course, which we can use for target identification or validation and to look at pathways in more detail. But often we will combine these capabilities in one service. So I'm going to show some examples of how we actually do that in practice and show the different stages of the drug discovery process. Now, this rather simple explanation of a linear explanation may not hold true for all development programs now, but I think is useful in describing some of the key areas where human data is helpful. At the earliest stages, often the fixed or frozen samples are used to identify a drug target in a diseased tissue. Beyond that, the fresh tissues can be used to validate pathways and targets. They can be used to select lead compounds or to investigate preclinical safety, or even to troubleshoot clinical observations. And quite often we are asked to give an explanation for unexpected side effects that have happened in the clinic. All of these activities together are de-risking the discovery process because we're generating more human data. We're then able to base decisions on human rather than animal data. So some examples of how we do that in practice, and picked a few different stages of drug discovery. Here's one from a target validation experiment where we're looking at healthy and diseased tissues, and we're comparing the effect of different pathways in those healthy and diseased tissues. We're also able to study various endpoints. Here we're looking at bronchoconstriction or dilation of isolated airways, but it could also be inflammation that we're looking at, for example. In the upper right panel, you'll see one way in which we control for variation between donors or between individual tissues is to conduct multiple replicates. So here we've done 8 replicates of tissues from that same donor, and we see very similar responses in those tissues. We will also then compare between patients and look at the variation between individuals. For many endpoints, for example, bronchodilation, the response in healthy individuals is very similar. However, when we come to diseased tissue, this is when we start to see that variation in response, very similar to a clinical trial. So human tissue data is certainly not as reproducible as the experiments in animal data. If we use 6 rats with a similar genetic background, whether inbred or outbred, we get the same response. That's fine, but it doesn't reflect the actual responses in the patient population. What we have here is a mini clinical trial, which is a snapshot of that patient population. So we do see more variation, but that variation between donors is actually giving an indication of how effective the drug will be in the target patient population. You can see here in the upper right panel, we've now have a graph where we've looked at the response to a standard compound in 3 different patients. And here you can see very similar responses in those patients. So for certain pathways in healthy tissue, they may be similar. For diseased tissue, we look at the actual target patient group, we may see differences. Moving on a stage to selection of lead candidates. These are probably the most common types of study that we do is a lead optimization, where we have relatively small number of candidates. We're not screening here. It's not high throughput screening by any means. It's secondary or tertiary screening or verification of efficacy in small numbers of lead candidates. So for example, here we are using mucosa of the human intestine. We're looking at the magnitude of response of different lead candidates in being able to increase fluid secretion in the gut. And we can directly compare those compounds and we can rank those lead compounds according to their efficacy in humans. We can also compare their responses to standard of care compounds. So it gives a very powerful head-to-head comparison of your lead compound to standard of care compound. We may also use these types of tissues from the gut to look at drug absorption, because ultimately the efficacy of a compound may not only be determined by its response when it engages the target receptor, but it's also determined by its bioavailability. This is something which we can investigate in fresh tissues using mucosa set up in an oozing chamber. Being able to study this in different individuals gives us an indication of the gastrointestinal bioavailability. I'll come back to this later on because some very interesting studies published by AstraZeneca and others has shown how the oozing chamber, which is named after a German physiologist, Hans Oosing. The oozing chamber is a really useful method to look at clearance, not only absorption, but also metabolism and any active transport that may prevent a compound from being efficacious by limiting its bioavailability. And then, as I mentioned earlier, preclinical safety. We saw the small video of the heart earlier. That is often used to look at the side effect profile of potential drugs by assessing whether they are going to impact cardiac output, either increase or decrease cardiac output. Neither of those are something you want to see as a side effect. We can also measure responses in blood vessels or airways or gut as off-target effects. So here we've got that response of a small piece of cardiac muscle. We're looking at the increase in force. In response to a control drug here is just isoprenaline. And you can see that increase in force, which would translate to an increase in cardiac output. These types of studies that I've shown are used by many companies. We've worked with over 150 pharma and biotech companies over the past 16 years. I don't have time to go through all of the many types of studies we've done. So please feel free to contact me about any particular interest. But I thought I would give a snapshot of some different areas of how human tissue has been used in different stages of drug discovery, from target validation through to safety. Here we have studies done by Proteon Therapeutics, which were around validation of a target for atherosclerosis. Using human fresh atherosclerotic arteries, we were able to look at the exposure of the living tissue to the test drug, which was designed to degrade elastin and permanently dilate those vessels. We conducted these target validation studies, which led on to preliminary dose finding and actually helped to inform future clinical strategies. With Nuraxon, one of our first clients that we worked with, we did many studies to investigate the potential side effects of a new treatment for migraine, which was targeting neuronal nitric oxide synthase. There were concerns that an off-target effect could be to inhibit endothelial nitric oxide synthase, which would not be desirable as it could cause an adverse increase in blood pressure. So we wanted to look at different compounds and show that they were selective for neuronal nitric oxide synthase and were not affecting endothelial version. We were able to do that by taking tiny arteries. These human small arteries you can see in the image are key to the control of blood pressure. These small arteries are the site where your minute-by-minute blood flow to specific organs is controlled. It's the constriction or dilation of these arteries that regulates blood flow to the skin or the muscles or the gut. And in total, they control your vascular component of blood pressure. So any change, even a small change in the control of these arteries, could lead to a risk of an increase in blood pressure. We were able to look at different compounds and how they dilated and also influenced constriction of these small arteries. And so we had a functional demonstration that ran alongside other tests that were being done on enzyme inhibition. So it strengthened the package of data to show that these compounds were safe and to support the selection of a lead compound. Merck also took this a stage later in looking at drugs that were already on the market, but for which there was a known side effect on blood pressure. Rather than raising blood pressure, they looked at two drugs that had known risks of hypotension, vernaculant, an antiarrhythmic drug, and flecainide. These had both been known to cause hypertension, but without any clear mechanism of action. We tested the effects of both drugs in both those tiny arteries I showed in the previous slide, and also the beating cardiac muscle that I showed in the video earlier, to look at two possible mechanisms of the hypertension. We were able to rule out in vitro any effect on the small arteries or the cardiac beating by vernaculant, but flecainide did reduce the force of the cardiac contractions. So this was a very useful study in explaining the potential mechanisms of these drugs. So even for drugs which are in the clinic or even on the market, human data can be used to add further value. There's a range of publications. I'm not going to talk through these. Again, if anyone is interested, we're very pleased to provide you with links to these. Similarly, there are some good review articles by ourselves and others, including a fairly recent book which has contributions from many human tissue researchers. We have one chapter on fresh tissues in here, published by the Royal Society of Chemistry. So there are some good review articles if you're interested in seeing other examples of how human fresh tissue has been used. So in the other two sections of the talk, I would like to move on to the regulatory environment and human tissue studies. I will start by summarizing a working party's efforts that led to a useful survey of the industry published in Nature Reviews, which highlighted the ways in which big pharma, CROs, biotechs are using human tissue. In particular, their use of human tissue for safety assessment, because that's the key interest of the regulators being in safety and tox assessment. You see the link to the publication here by Anthony Holmes of NCCRs, Frank Bonner, who's experienced toxicologist, and David Jones is the MHRA, the UK regulators, which describes assessment of drug safety in human tissues. What are the barriers? What are the ways in which pharma would see the future use of human tissue? So I've very quickly summarized some of the key findings, which are quite interesting. One of the first questions asked was about the current use, and it found that roughly 50% of big pharma, there's 27 pharma companies, so big pharma were providing one submission each, about half of them using human tissue-based assays. However, the majority were evaluating them, and the vast majority of them were considering them in future use. So there seemed to be a trend towards increased use, with roughly half of them using the systems at present. The main reasons that they were doing so was overwhelmingly about human tissue models being more relevant than other systems. Other reasons given were around deficiencies in animal models. Drilling down a little bit into the types of assays and studies that, excuse me, that pharma are actually doing. In the survey, they were given a number of choices between fresh tissue models, such as tissue slices, and ex vivo tissue preparations. So that's the type of tissues that we've been talking about today. And also other systems in 3D culture or 2D culture. So you can see a number of approaches been taken by pharma, but the biggest use is in ex vivo tissue preps. Part of this will be driven by the fit with the regulatory guidelines, ICH S7A being the most relevant one on safety pharmacology, but other guidelines and biologicals also playing a role. Now, none of the human fresh tissues experiments are mandatory. That's unsurprising given the historical reliance on in vivo models and cross-species comparisons in animals. But the regulations do make specific mention of human data, and they do make specific mention of tests, the types of which we have talked about today, and we'll give some further examples of. So I thought this is a useful summary as to the actual points in the regulatory guidance that specifically mention endpoints as measured in fresh tissues. I'm going to pick just one of them today around the lung, because we've already talked about cardiovascular. Then I thought I'd give an example of respiratory safety assessment. And firstly, to ask the question, to do a regulatory test, are there actually enough tissues out there? Well, there certainly are enough. There may be challenges in acquiring those tissues from ethics, logistics, having the correct networks, and that's really why CROs are best placed to do this type of work. However, the tissues do exist, and patients are very happy to donate tissues for research when it's made very clear about the commercial research and the benefits longer term, not to them directly, but to patients in general for the conditions that they may be suffering from. So there are huge numbers of organs available for transplant, many of them referred for research also, and also similarly high numbers of surgical residual materials. These can be used by safety pharmacologists to look at the excuse me, unexpected observations during the clinical studies, to compare species, perhaps in direct request from regulators. But I'm going to also pick up on this final point about how the tests we do in human fresh tissues sit with the S7A guidelines. Now, those guidelines describe tests that should be done on pulmonary ventilation, and they aim to understand the effects of the drug on breathing. So, what endpoints do we actually consider when we're looking at the safety of a drug in relation to the respiratory system? Well, we need to consider whether the drug interferes with the respiratory muscles, any central nervous system control, or chemo or mechanoreceptor control of the ventilatory apparatus, how we're moving air in and out of the lungs. And we also need to consider how the gas exchange units are affected. So any effect on the airways, the alveoli, the pulmonary vasculature, or parenchyma, which can affect gas exchange. And this may be through effects on things like airway resistance or lung compliance. So these two broad areas are things to consider in terms of safety. The main focus of the guidelines, however, are on the ventilatory apparatus. In fact, when we look at the failure rate, the failure rates for different problems, either in ventilation or the gas exchange unit, don't quite match the guidelines. So when we look at the problems, this is an analysis conducted by Dennis Murphy at GSK, and we look at the left-hand bar graph for problems due to safety issues in respiratory system, which have been reported in clinical trials. We see problems due to airway obstruction, breathing abnormalities, or lower respiratory tract abnormalities. And if we plot them as a percentage, you see they're pretty equal, really, in each of those three areas. But you see that the ventilation, breathing abnormalities, is only one part of the problem. That's the part that guidelines mainly focus on. However, we also look at the problems and where trials have been stopped or there's been issues in the clinic. They are often attributed to the gas exchange unit. Now here the guidelines say they should be considered and by secondary tests in vitro. So these portions in red actually account for around 60% of the clinical trial problems due to respiratory issues. So we can measure things like airway resistance or lung compliance, but why don't we simply do that in animals as well? Although the regulations focus on in vitro, in vivo animal models for ventilation assessment, why don't we try and tackle the rest of it through in vitro animal models? Well, the problem is when we look at responses in vitro, we don't see the same as in humans. And in humans, we can look at bronchoconstriction quite readily in these lung tissues that we obtain from transplant or from surgery. If we look at the responses of human versus the main preclinical species, here we've got some rat trachea bronchi looking at responses to acetylcholine or serotonin. So these are bronchoconstriction responses to these spasmogens. In the human bronchi, we see actually no response to serotonin. So the rat is completely hopeless as a model. Of course, we could use other species. However, if we plot other species in terms of their EC50 bronchoconstriction concentration, then we plot that against different bronchoconstrictors, methacholine, histamine, and so on. And then we plot that against the main preclinical species and add in human EC50s. We see that no animal model closely reflects the human condition. So there simply isn't an animal model that is going to predict that in vitro response to the effects on the gas exchange unit. So if we want to reduce the number of failures in the clinic that we saw from Dennis Murphy's paper, then we're going to have to consider approaches in humans. And that takes me on to the last section of species differences. So we've started to touch on some examples there in the lung, but it's not only in the lung that we see differences between human tissue and animal tissue. And it's certainly not the case that we would look at animals and tissues and say it's not of any use. It certainly is valuable to research and may, in 60–70% of the cases, may well reflect the responses that we see in humans. But in many cases, we don't see a similar response. We talked about serotonin in the lung. Well, here are the effects of serotonin, also known as 5-HT or 5-hydroxytryptamine. Here are the responses in coronary arteries and subcutaneous arteries. So these small arteries that control blood flow, we see really small responses in dog, which is one of the main species used in cardiovascular safety assessment. However, we see a much bigger response in human. So using dogs to model responses of serotonin agonists could lead to false negatives when predicting human safety. Here's another example in canine and human arteries, and this time with a phosphodiesterase inhibitor, PDE2 inhibitor. And again, we're seeing a less sensitive response in the canine arteries than we would in human. So again, the canine, if we use that as a model of safety, we would underestimate the potential for risk, and we could see a side effect in humans that's not apparent in dogs. Moving on to bioavailability, giving some examples there of species differences in the lung and also in cardiovascular system. We also see differences in how drugs are absorbed and metabolized in different species. So looking at the measurement of drug absorption in the gut, the rats and humans' oral absorption of drugs is actually pretty similar. However, the metabolic activity, metabolic enzyme activity in the GI tract is very different. And so we have to be careful that when we look at just straightforward absorption of a drug, passive absorption across a membrane may be the same. But once we consider total bioavailability, which is also affected by the presence of transporters that may actively efflux drugs or metabolic enzymes that could break the drug down before it actually is absorbed into the bloodstream, then we can see a quite different picture. And the graph on the right shows what's quite a messy graph, published by Grass and Cinco some time ago now, comparing human bioavailability to animal bioavailability. And really the point of that graph is to show there is no good relationship between the two, between human, rodents, dogs or primates. To give a couple of more examples why that is the case, if we look at dogs and humans, their oral absorption is different, so the human bioavailability is not well predicted. In monkeys versus humans, this time oral absorption is pretty similar, so passive absorption across the mucosal lining is similar. But again, like with the rat, the metabolic enzyme activity differs, and so we see very different bioavailability. We're not able to predict well based on use of the non-human primates. So I mentioned earlier the publication by AstraZeneca, who took all of the data that they had collated over around 15–16 years in studies in human fresh gastrointestinal tissues, and also published data. And they looked at the correlation between human absorption in vitro, human drug absorption in vitro using fresh gastrointestinal tissue, and actual bioavailability, and they found a much better relationship between the fraction of drug that's actually absorbed and passes through the gut and would get into the bloodstream, and what can be measured in the oozing chamber using a section of fresh small intestine. Again, I've included some example data from the AstraZeneca paper, and in the upper right, data generated at REPROCELL using fresh small intestine. So to finish off, and before we take any questions, to summarize, human tissues are being used to add commercial value by showing human proof of concept, also to reduce the risk of clinical failure. Having the efficacy and safety data generated in humans avoids these issues with species differences. That can be used all the way through the drug process, and regulators are quite open, and in fact in the UK at least are very encouraging towards the use of appropriate tests that generate data in fresh tissues. So thanks very much for attending, and also I'd like to open up to any questions.
Thank you very much, David. I would just like to remind our audience that they can submit questions using the Q&A function. There's a button located at the bottom of your webinar window that you can use to submit questions. A few questions to start out with. David, can you talk a little bit about positive and negative controls? How do you find them?
Mm-hmm. Sure. Yeah, so typically when we design a study, it's done in partnership with the client, and so the scientists in both groups will get together and decide what is the question that we're trying to answer, and that will dictate the selection of the relevant positive and negative controls. And that positive control may be a standard of care compound, or it may be a standard agonist, such as what we showed with the bronchoconstrictor agents, hormones or neurotransmitters for which there is known concentration response data. The negative control, similarly, it may be a compound for which there's a known absence of an effect on a pathway of interest. So what we do find with human tissue projects is they are highly customized. Each project tends to be designed for the specific requirements and scientific questions that are being asked.
Okay, thank you. There's a few questions about the tissues. For the tissues, do you have access to any of the clinical data for the donor of the tissue?
Yeah, that's a good point. Yeah, I didn't mention that, and we do, and it's becoming very important that we do. We obtain standard medical information in an anonymized form, so we don't know the identity of the patient, but we do have access to their medical history. We also are in the system in the UK, we're able to do longitudinal follow-up, so we can request clinical data for patients to look at outcomes further down the line. And that clinical information is becoming very important for precision medicine, where we may see a greater effect of a test drug in a particular group of patients with certain clinical history in comparison to others. And I briefly mentioned our ability at REPROCELL to do gene sequencing or transcriptomics. And so we have recently linked the response that we measure in a fresh tissue to the genotype of patients, so we can compare how drug responses change based on that clinical history and also genotype or transcriptomics.
Okay, thank you. And then how are the tissues treated between the time that they're collected from the patient and the time that you start the experiments in the lab?
In a very similar way to transplant organs. So we have a time window, we have very careful training of the sites and protocols that they follow for transport solutions, for temperature during transport. We have specialist couriers. So it's a very similar process to that used for transplantation.
Okay, and is there a kind of time limit that you need to get it to the lab?
It varies between tissue types. So I gave an example of the heart tissue. I guess most people will be aware for transplanting a heart, it's very time critical. We have in a matter of hours to get it to the lab and start to dissect that tissue or even use that tissue in experiments. Other tissues will survive a little longer and can a bit more robust, like skin tissue is a bit more robust. So it really depends on the tissue type, but what's important for us is that we have all the logistics in place and those rapid transport of tissues to the lab during the night, at weekends. Our staff are here handling those tissues.
Okay. So are all of these studies done in the lab in Glasgow, or do you have other facilities and some of the other REPROCELL facilities that also do these type of assays?
Yeah, so we have labs in Beltsville in Maryland and also in Glasgow in UK. Both of those labs do the same things. We have access to tissues in both US and UK. And the choice of where we do the work is usually dictated by where we source the tissue. It's the same quality system, the same training, the same procedures. We have our lab close to the tissue sites so that we can, for the reasons you asked, we can get the tissue quickly and we can do those experiments.
Okay. And then what would the process be if the customer comes to you and wants to do an assay that's similar to what you've done before, but using, let's say, a different tissue type? What would you do for a validation for that?
Yeah, that's a very common occurrence because there are so many different types of tissues we can get and many targets, many different types of endpoints. So we are often doing small pilot experiments to establish activation of the pathway of interest and make sure, perhaps with a standard of care compound or a positive control, make sure that we can engage the target, we can measure the endpoint, and do that in a cost-effective way as a small pilot before then doing the main study.
Okay. Are there any other questions from the audience? Okay, I think that's it. Thank you very much, David, for your talk. And please, if you have any questions about the webinar, you can send them to me in the email address that you got in the registration in the invitation. This webinar has been recorded and will be available on our website in a few days. Thank you very much.
Thank you, everyone.